Hi everyone, good morning. My name is Amber Carter. I'm a genetic counselor at INVITAE. I'm also a clinical program manager for our somatic assays in the sort of research study side of things. Green arrow? Okay. My disclosures are that I am an employee and shareholder of INVITAE. So we'll go through a quick refresher on circulating tumor DNA and MRD in case that's not something you're dealing with on a regular basis, and go through the clinical dilemmas that are really prompting the needs for ctDNA-based MRD assays. I'll talk a little bit about INVITAE's personalized cancer monitoring, or PCM assay, and we'll talk about some of the emerging data in different tumor types. Sorry, I'm really bad at pressing this green arrow. Here we go. So just to set the stage, cell-free DNA, I'm sure many people in the room are familiar with it, but it is fragments of DNA that have broken free of the nucleus. Most often comes from dying white blood cells, and it is made up of fragments wrapped around histones in about 20 base pair segments. And on average, that fragment length is in the ballpark of 167 base pairs. It also has a very short half-life. It degrades very quickly. Circulating tumor DNA, if a patient has cancer, there may be DNA that has also broken free of the nucleus from tumor cells and made its way into the circulation. And so ctDNA is a portion of the cell-free DNA in patients who have cancer. It is typically a shorter fragment than the background cell-free DNA. So ctDNA is actually one of many tumor markers floating around in the blood. And another one that you hear a lot about recently are circulating tumor cells. That sounds very similar, but these are two different entities and similar but different uses and assays. Circulating tumor DNA can also be pulled out of other fluids like urine, cerebrospinal fluid. There's a lot of cool uses for it. Tumors will shed varying amounts of ctDNA depending on the tumor type, the histology type, the stage, whether or not the patient's undergoing treatment. So it's quite variable. However, it is typically quite low. So ctDNA is usually present at 1% or less in a patient with cancer, 1% allele frequency or less. So we also know that ctDNA relates to tumor burden, as in tumor volume, and higher VAF of ctDNA would relate to higher tumor volume. So there's many ways to use ctDNA in a patient's cancer journey, but in patients who already are known to have cancer, we're gonna focus in on minimal residual disease, or MRD. So MRD refers to the evidence that there is presence of residual tumor cells after some sort of curative intent therapy that cannot otherwise be detected by traditional methods, right? So like imaging. You might also hear the term molecular residual disease, and that just refers to the means of detection. So in this diagram, you can see an example of thresholds with those dotted lines, or hopefully you can see them. And so there's different thresholds for how much tumor volume needs to be present for something to show up on a scan or something to be detected by ctDNA. And those are typically different thresholds. So it's frequent that an assay will pick up evidence of ctDNA before evidence of the disease can be picked up on CT scan. So I'm gonna be talking about MRD a lot, and that's the main catchphrase lately, but really MRD tests in solid tumors go beyond just detecting whether or not it's present. So for example, if you're thinking about a situation where a patient was not having surgery because it was a metastatic disease, and so you fully expect there to be plenty of ctDNA present, you can also use these types of assays as a way to assess therapy response. So looking at increasing or decreasing levels of ctDNA. Over the past decade, there have been lots of different approaches used for detecting MRD. And It's ranged from sequencing efforts from PCR to next generation sequencing, but really sort of the philosophy for how you're trying to detect MRD falls into two main buckets, either tumor agnostic or tumor informed. Starting with tumor agnostic, these types of assays are a fixed panel, typically going after common alterations. It might be common alterations in one tumor type, like breast cancer, or common alterations in multiple tumor types. And then tumor-informed are assays that rely on the specific genomic alterations in a patient's tumor in order to create the assay, so they're bespoke to each patient. So there are advantages and disadvantages, right? So the tumor agnostic has the advantage of not requiring tissue, which can of course be a problem when you're trying to track that down or there isn't enough of it. But tumor informed typically has the advantage of higher sensitivity. So just to give you an example of how a tumor-informed approach begins, it requires a sample of blood like you would take for hereditary testing in a purple top tube, and it requires FFPE tissue typically. There are some methods that can be done on fresh frozen, but FFPE is the most common. So this is an example of INVITAE PCM requirements. It has to start with blood and tissue, and that's to create the first part of the assay. And then to actually test for ctDNA, you require blood that you can extract cell-free DNA from. And so since that circulating tumor DNA is going to degrade so quickly, it has to be preserved in certain types of tubes that are going to allow it to get from the patient to a laboratory. And then that last step can be repeated over and over again. So what happens to create the test is it starts off with a germline whole exome and a tumor whole exome. And then based off of those results, in our case, we use a proprietary algorithm to select variants. They're really being selected based on, high confidence, low noisy regions, making sure it's not germline, ruling out CHIP, that sort of thing. And that creates a patient-specific panel of up to 50 variants that have been curated for that patient. And that's going to follow that patient over time. If they were to have a test today before they have treatment, and then they're going to have it again, say, in 6 months or a year, it's the same panel that's going to be leveraged for that patient. So then we take the cell-free DNA and apply that panel to it and get an MRD call. And so that's a positive or negative call or a percentage, and that comes out as a report. So that last step can be repeated over and over again. So we do select 18 to 50 variants because that's where we have been able to maximize our sensitivity and specificity. It also gives us flexibility for patients with higher or lower mutational burden, right? So that we're able to actually create a panel for most patients, but optimizing sensitivity. And as far as the levels of sensitivity go, the analytical validation showed better than 99.9% down to 0.008% allele frequency. And that is important because just the overall cancer population is going to have such low levels, particularly early stage tumors, and particularly after they've already had treatment. They're going to have really low levels of cell-free DNA. And that's one of the ways we leverage our Archer anchored multiplex PCR or AMP chemistry, because it selects for those smaller fragments that really are the circulating tumor DNA within the cell-free DNA. And our specificity remained high down to 18 variants. So moving on to potential clinical dilemmas that ctDNA MRD assays can help with. So risk stratification, either before treatment or after treatment, picking up evidence of relapse, so in the surveillance period after a patient's curative intent therapy, and also in terms of therapy response for various stages of cancer. So clinical dilemma number one is just risk stratification. So it's a well-known fact that patients are different risk levels after they've had surgery for a cancer, right, an early stage cancer. But what we don't know is who is higher risk or lower risk. And so large groups of patients tend to get adjuvant chemotherapy where a small portion of that group is actually benefiting from it. So some patients were cured by surgery. In fact, most early stage patients are cured by surgery alone, but we don't know who they are. And then some patients just choose to forego adjuvant chemotherapy because of the toxicity, but maybe they actually needed it. They would have benefited from it, but we don't know who they are. And then there are patients who go through surgery, adjuvant chemotherapy, and there's still evidence of circulating tumor DNA, but we don't know that because we're not testing them, right? But they could benefit from potentially more therapy or clinical trial access, that sort of thing. So the common thread here is there's varying risk levels, there's varying things that could be considered based on those risk levels, but we have to identify who those patients are. And providers are already using risk stratification in their calculation of who to recommend adjuvant chemotherapy for. They already use clinical pathologic features, traditional biomarkers, like this is an example of stage 2 colon cancer, so CEA. But this is one more piece of evidence, since we know that if there is ctDNA present, they are at a higher risk of relapse because there are remaining tumor cells. This is one more piece of information to pull into that risk stratification model. The next clinical dilemma is just trying to detect relapse before it happens, or at least before there are clinical symptoms. So this is sort of a constant lingering concern for any patient with cancer, right? That you've had your therapy and now you're in the surveillance phase. And we're kind of stuck waiting on patient symptoms, imaging to show macroscopic disease, or CEA, CA125, those sorts of biomarkers. And so there's a lot left to be desired just as far as finding this sooner, preferably before an early stage tumor has actually come back and metastasized. Going back to this picture I showed you earlier, if you follow that yellow line that goes through the middle, you can see in that patient hypothetical example that the ctDNA threshold is hit before the CT scan threshold. So that difference concept is called a lead time. And we see that in a lot of studies that ctDNA picks up relapse ahead of imaging studies. Now, even in patients who maybe were ctDNA negative right after surgery or right after therapy, and that was reassuring, we know that sometimes those levels are just too low right after therapy, that they can fly under the radar and not be detected. So what many studies have demonstrated is that if you follow patients over time with ctDNA-based MRD testing, you're going to increase your sensitivity of picking up those patients who are at that much higher risk of relapse. So when you look at MRD literature in solid tumors, you see some themes in the time points that are being used. And so one is often referred to as the landmark time point. That refers to a time point shortly after therapy. So either shortly after surgery, shortly after adjuvant therapy, usually in the ballpark of a couple of weeks to 3 months. And that sort of sets that landmark that we can go back to and say, if a patient tests positive or negative at that time point, what ends up being their risk of recurrence? The other time points that you see a lot are surveillance. They might be called serial or longitudinal. Those will be at different cadences in different studies. There is no agreed upon frequency of those yet. But all studies thus far have been consistent that the serial time points increase sensitivity beyond the landmark time point. The other time point that is often used is a baseline before surgery, and that can just be helpful in determining sort of biologically, is that tumor shedding a lot or not from the get-go before surgery even happens? So lastly, I have a handful of studies I thought you might be interested in, specifically in lung, breast, and colon cancers. These are using various different assays with the consistent theme that they are all ctDNA-based detection of MRD. The first one is one of our studies. This is in lung cancer, specifically early stage lung cancer, and it was a collaboration between Archer, INVITAE, and Cancer Research UK and the University College London. These patients were followed for at least 5 years unless they left the study or passed away. So this was an earlier iteration of PCM. Patients were tested at time points before surgery, in that landmark time point after surgery up to about 3 months, and then periodically every 3 months for the first year or so, and then moving into more of a slower cadence every 6 months. They also had their standard of care imaging, and that did vary by site. So the findings from the first group that was reported, these were 78 patients tested with PCM, was that in the patients who clinically relapsed by that point, ctDNA had been detected at or before evidence of relapse in 37 out of 45 patients. And there was a whole range of lead times, as you might imagine, with all those different sort of standard of care variable methods of using imaging in clinic, but the median lead time was about 5 months. No ctDNA was detected in most of the patients who had not yet shown any evidence of clinical relapse. There was one patient out of 23, one sample out of that patient, that did come back as positive, and they had still not shown any evidence of relapse. And then in patients who, they looked right down at patients who had developed a second tumor to see if the test sort of accidentally flagged that tumor, and they didn't. So 10 patients had second primary tumors, and remember that the assay is built off of a specific tumor, and no ctDNA was detected when you were just looking for the genomic alterations from that first tumor in 10 out of 10 patients with a second primary. And based on this, preliminary results of this study, a couple of interventional MRD studies were generated looking at how can you use this test to then direct care and either sticking with standard of care or adding in, say, immunotherapy in patients who are ctDNA positive and at higher risk. This next study is very similar to the last one. This is from an academic group that enrolled a little over 100 patients with early stage non-small cell lung cancer. They looked at similar things as the last study and very, very consistent results. But what I wanted to show you is one thing they did looking at whether or not patients received adjuvant chemotherapy. So sort of thinking, how can we backtrack this information to lead to more clinical trials and how to use the test? So in stage 2 and 3 lung cancer, patients typically do get adjuvant chemotherapy, but there's, again, kind of minimal benefit to the group as a whole. So it's not uncommon for patients to just decline it due to toxicity. So the investigators went back and looked at the patients who were ctDNA positive or negative and whether or not they had gone on adjuvant chemotherapy, and then teased it out based on that landmark time point in this Kaplan-Meier curve. So the two lines that end up closest to the top, they're green and blue, they are patients who are ctDNA negative. Those separate out nicely from the red and black lines at the bottom, patients that were ctDNA positive, so not surprising at that landmark time point. If you were negative, you had a better recurrence-free survival than if you were positive. But then if you look closely at the two lines towards the bottom, apologies for the red, that's the second one from the bottom, and the black line, they clearly separate. So that's suggesting that patients who were ctDNA positive benefited from the adjuvant chemotherapy. So this set the stage nicely for some interventional trials in this type of cohort. This is one of the first and larger studies of MRD in breast cancer. This is from the Institute for Cancer Research in the UK. They collected plasma from patients for quite a while, and honestly quite early on for the field. It's pretty impressive. They recruited 170 women across breast subtypes, and they followed them every 3 months, and then for the first couple of years, and then every 6 months. So if you look at the Kaplan-Meier curve all the way to your left, those are the patients with ER-positive breast cancer. And that blue line all the way across the top of the KM curve, those are the patients who tested ctDNA negative at the landmark time point, and within 5 years, none of them recurred. Now, ER-positive breast cancer does have late recurrences in that like 5 to 10 year range, but it does show that if you were negative at the landmark time point, your risk of recurring in 5 years was exceptionally low. Now, that did not hold true for ERBB2 or HER2 and triple-negative breast cancer. I should say they were very low, but there were patients who were negative at that landmark time point who did recur within 5 years. So that just is a good reminder or gut check for all of us in the field that a landmark time point is not perfect, and we need to think about ctDNA that may have been too low to detect at that time. But consistently across these different subtypes of breast cancer, there's clear separation of the curves, and that landmark stratification really did separate out prognostically who was higher risk of relapse. Moving on to colon cancer. Colon cancer has a lot of active research into different clinical dilemmas where ctDNA-based MRD tests can be helpful. In this one, about 240 patients with stage 2 or 3 colon cancer were tested, and at that landmark time point, it also served as a good way to risk stratify patients. So if the patient was ctDNA positive at that landmark time point, their recurrence-free survival was only 39%. But if they were negative, they had a low recurrence risk. Their recurrence-free survival was 89.4%. And then in that same study, the investigators looked at how does adjuvant chemotherapy help the patients that were ctDNA positive, and can you derive information from the ctDNA after adjuvant chemotherapy to assess the response? So in this event chart, I know it's probably hard to see, but essentially there are 5 patients who were positive after their surgery. They went on adjuvant chemotherapy, they were tested again, and they were negative, so they cleared their ctDNA. And all 5 of those patients continued to be ctDNA negative over almost 2 years and were still relapse-free at the time they collected the data. Got it, thank you. And then getting back to those surveillance time points, we know from a lot of studies that they really can detect relapse ahead of time. But in this same study, what you're looking at here is a graph of patients who tested positive for ctDNA at least one time point after their adjuvant chemotherapy. And so it showed that those patients, it's really combining a lot of time there, but those patients had a hazard ratio of almost 13 that they would relapse. In this study and many others, ctDNA has been lined up next to CEA, which is used very commonly in standard of care. It's in the guidelines. And ctDNA consistently complements CEA, going beyond what it can do. But the two of them seem to work together nicely in the clinical picture. It doesn't want to let me advance. There we go, just a delay. So what do we do with all this information? These tests are clinically available from multiple labs. They're out there as RUO tests as well. Patients are asking for them, providers are interested in them, but they are not in the guidelines yet. And we know from lots of other emerging technologies that takes time, and every clinical scenario may be different, not to mention every tumor type. So we need a lot of studies. We need escalation studies based on ctDNA positivity or negativity, or de-escalation based on ctDNA negativity. And we're really excited about working on a lot of those studies in our group. And lastly, I would just invite you all to come visit us at the booth if you have any questions about this, or reach out if you want to talk more about collaborations. Thank you so much for your time and attention.